Tysabri had the unfortunate happening about, say, 2 years into treatment, some people develop a fatal central nervous system demyelinating disease called progressive multifocal leukoencephalopathy, or PML. It's a whole lot easier to remember. So the FDA took Tysabri off of the market at that stage, reviewed the safety, came back a year later saying that the risk of developing PML, that the efficacy outweighed the risk. But it's something, the point that I make with all of these drugs, including the new oral medication, is that they come with degrees of risk. The side effects can make these drugs very, very, very risky. One hopes, of course, especially with the new oral medication, that this will treat me without putting me at risk for developing serious side effects, but unfortunately it's not the case. So that's something that you have to consider when taking these drugs, and of course your neurologist will talk with you at great length about. But the biggest frontier that I think that I know most about, and it's what I'm actively involved in, is the question about repairing chronic damage to the brain. So as a preamble to this, and it's important no matter how much you know about MS, myelin, the target organ, you get this quick reminder about the structure that we're trying to repair. So just a few slides to give you the background on what myelin is and why we might want to repair it. So the core of the brain, this is a brain that's been stained blue, is myelin. As you can tell, it's a fairly significant part of the volume of the brain itself. And if we take that blue stuff, as it were, and look at it under a microscope, we see that there are lots of little blue circles, and these blue circles are the myelin sheaths around individual nerve fibers. And then if we increase the magnification, you see 3 nerve fibers, 1, 2, 3, with a black ring around them. That's the myelin sheath, and that's what's essential. Some of you have heard me talk before, and nonetheless it's called reinforcement. This black stuff is the myelin sheath. It's a membrane. It's very rich in lipids or fat, so it's a good insulator, and you need this to perform normally. It's like an electric cable or a telephone cable. It must have its surrounding coat to work properly. It's produced by a cell. This myelin membrane, it's not the nerve substance, it's produced by the cell called the oligodendrocyte. There's a spelling test at the end, by the way, so write it down. Oligodendrocyte. Split it up with that. So this is the cell that we need to make the myelin sheaths, and this is in development. Here we see one cell that's making these 3 dark rings of the myelin sheaths associated with it. And if you take that tissue out and put it into tissue culture in a dish in the lab, as it were, and look at it stained with markers for the individual cell types. Here we see what they are. The oligodendrocyte in red is labeled with this red antibody, as it were. And then there's another very important cell in the nervous system, essential for remyelination, called the astrocyte. So these are the cells that work together to make the myelin sheath in development. Complex slide, but you need to know this. So I was trying to think of an analogy of this morning while trimming the talk a little bit. So when you are born and you grow up, you're a baby, and then you become an adult, you look very different as you grow older. And the same happens with the oligodendrocyte. All of the cells in our body, of course, arise from the mother of all cells, called the embryonic stem cell, or the ES cell. So like the baby, as it were, my analogy, maybe it's not a good analogy, but it's the only one I could think of. The cell changes as it grows older. And as it grows older, it goes through different stages. It's called a progenitor, the oligodendrocyte progenitor. And then it becomes a mature cell, and the mature cell is what we need to both make the myelin sheath and to maintain it. And we study this. This has been the subject of, I don't know, hundreds of papers in scientific literature. Huge amounts of time and effort to study this cell. It's really important to know this, because we can know how to replace the cells, how they get damaged, how we can make them divide, migrate, etc. So the more we know about the biology of the cell, we know how to repair it. So that's the background. In MS, what happens is you lose myelin sheaths. So the red, or the green arrows rather, indicate large areas of myelin loss, called plaques, in the brain of someone who's had the MS for many years. The red arrows indicate sort of slightly less dark blue areas. What these are areas that actually have been repaired. They're called shadow plaques. And in fact, what they indicate is the ability of the brain to remyelinate itself. So we actually have self-repair capabilities. The problem in MS is that eventually that self-repair mechanism dies out and you end up with chronic persistent damage. Now, for 5 years at UW, this has been our target. It remains our target at the end of our big MS grant. So our thesis is the notion that there is a subpopulation of people with MS who have an area of demyelination in the spinal cord in particular, strategically placed. If you have damage in the spinal cord, it's likely to lead to clinical symptoms. You could have an area like that in your brain and be completely asymptomatic, i.e. it's a silent lesion. This MRI targets a big lesion in the spinal cord, and this person is very likely to be in a wheelchair, to be paraplegic, probably have problems with their arms as well. So the question is, this is the site that's causing all the clinical dysfunction. How can you repair it? And that's been our target. So this is what it might look like looking at the microscope. You see the white area is the loss of myelin in two parts, a big area of loss of myelin. And if you look at the higher power under the microscope at this, now you see the white structures are the nerve fibers, but they don't have the blue surrounding coating. In other words, they are demyelinated axons.